general definition. And we're going to find in just a moment that there are several basic chemical classes of hormone that you must learn. You must learn the chemical classes because the chemical class of the hormone will tell you the hormone's mechanism of action. It will tell you how and where it binds. And when you learn the chemical class of the hormone, you will also know how the hormone is transported. So the general principle, and we talked about this before, is that you're pretty much a sack of water, right? You're just big sacks of water. And your blood is chiefly aqueous. But your cells are surrounded by a hydrophobic membrane. It's kind of analogous to the oily film that you see on the surface of the ocean or something. See when a boat leaves its trail of gasoline or hydrocarbon. You see it as that rainbow or chromatic pattern on the surface of the water. In many respects, then, we're just sacks of water that are compartmentalized by these hydrophobic membranes. All right, that presents a problem because depending upon the structural class of the hormone, some hormones access their receptors. Remember, hormone only works if it binds to a receptor. Some hormones bind their receptors at the cell membrane. All right, they travel through the blood and then bind to the cell membrane. Other hormones bind to receptors that are inside the cell. All right, so if that's true, we have a bit of a problem. If you were designing, I'm going to put you in charge of hormone design today. If you're designing a hormone, should your hormone be hydrophilic or hydrophobic? Louise says it depends on where you want it to go. I just want to get through the blood right now. So what should it be just to circulate through the blood? Hydrophilic. All right. What's going to be the problem if I design a hydrophobic hormone?
It's not on a protein.
Huh?
You have to put it on a protein.
I don't know what that means. We haven't got to that yet. I just want to. If we have a hydrophobic molecule in the blood, what does it do? It sort of comes out of solution. Right? And you have hydrophobic things in your blood right now. You basically are a jar of Italian salad dressing, right? You have. If you remain still long enough, if you sit at your desk like that long enough, what happens in Italian salad dressing when it's on the shelf? Yeah, it separates. The water goes to the bottom, it goes to the top. Yeah, all that. And so from time to time, you have to do this. You have to self-agitate to get the two mixed again. All right, why don't you have to do that? Because you do have not only hormones like hydrophobic hormones, but you have a variety of hydrophobic molecules. And I think in part the answer has been lead to you. You have in your blood molecules that will latch onto hydrophobic molecules. For example, what is a lipoprotein? There are HDLs and LDLs, high density and low density lipoproteins. What is a lipoprotein?
Binds to fat.
Protein lipid aggregation, but that protein enhances the solubility of that lipid in the blood. And this is a theme that will crop up again in our discussion of the reproductive system. We're going to encounter the same general phenomenon. But so usually it's a protein. Usually it's a protein that latches onto a hydrophobic molecule, some type of lipid, and the function of that protein is to act as a transporter or carrier of the hydrophobic molecule through the blood. So the point we're making here is that hormones can sometimes circulate free or bound to proteins. And really what determines whether they circulate free or bound to proteins is whether they're hydrophobic or hydrophilic. Remember we talked about the partition coefficient earlier. As a rule, peptide hormones and amines tend to circulate unbound. Everybody know what I mean or what I'm suggesting by unbound? They don't have a protein chaperone. By contrast, steroids and thyroid hormones, thyroids are going to give us a problem later, tend to require protein chaperones in order to be transported in the blood. So the question, Evan asked, does that mean that the steroid and thyroid hormones are, is that what you're asking? Are they hypertrophic? Yes, they would be hypertrophic as well. What is required in order for a hormone to induce a biological response?
Receptors?
Binding to a receptor. You see a problem here if you happen to be a bound hormone? Yeah, you're bound. You're already encumbered by a protein. So that means there must be some mechanism in place for you to detach your carrier and then bind to your receptor. So they have a number of other issues too. But for now, I just want you to be aware of this distinction, and also be aware that in fact it's a little more complicated than that. Some water-soluble hormones utilize carriers, and we'll talk about why that is a little later. So we've already seen that there are problems with our general rule, but I think now this is the general point that I want you to take home. Now that we understand this distinction, it is the free or the unbound hormone concentration that is physiologically important, because it's only the free or unbound, unbound hormone that can bind to the receptor. Of course, when hormone assays are done, and you'll be involved in interpreting quite a few of these, if not the tests themselves, most of those hormone assays, when you try to find out how much hormone an individual has, is it in a normal range or not? Those assays are for total hormone concentration. So what do we mean by total hormone concentration? Yeah, bound and unbound. And do you see how that might be misleading? All right, so just remember that what you're interested in, and of course now the computer printouts generally make this quite clear, but as a physiological principle, what you're interested in is the unbound or free hormone level, because it's that concentration of hormone that is available to bind to receptors. Most hormones, especially, we'll get back to this later, but most hormones are over time degraded and inactivated by either the liver or the kidneys. Sometimes this happens through what's called peripheral degradation at the target tissue. But remember, they typically have relatively short half-lives. We will see that the half-life of the hormone also is determined by its chemical structure. You can see we talk about serum half-lives, the time required to reduce the concentration in the blood by half. And then those metabolic products can sometimes be recycled, but they're generally excreted. And so this is just a little list of things I want you to think about. It says the interaction of the hormone with its receptors in the target tissue will produce a graded response influenced by hormone concentration. So we need to be aware of all the factors. You need to be aware of all the factors that might affect the concentration of hormone in your patients. Remember that the biological response is dependent upon the availability of receptors, and that concept is usually described as receptor number. We're going to need to talk about that a little more. What we're going to find is there's a relationship between 1 and 2, sometimes called receptor density. Remember, the hormones have a relatively short half-life. The receptors can be in a variety of locations. So this hormone receptor interaction is typically short-lived, but will determine the extent of the biological response. So we need to be aware of the factors that affect the ability of the hormone to bind the receptor, and to a large extent, it's going to be concentration driven. As long as both the hormone and receptor are present and available at sufficient concentrations, then one should anticipate the biological response will continue. That's what you would expect. Doesn't always work out that way. We'll talk about some of the reasons for that. It says here, interval between successive exposures would also affect the biological response. Why would that be? Anybody have any thoughts on that? The interval between successive exposures to the hormone. What would that have to do with your ability to respond to the hormone?
Is it similar to like a refractive period that needs time for it to um, have the gate, I mean, whatever, the receptor to be prepared to receive that hormone again?
Let me think about that a second. So you're saying there's a property or characteristic of the receptor that would be analogous to a refractive period. I don't, strictly speaking, I can't quite get to that, although I think you're on the right track. I don't think it's the receptor itself.
Is it like the concentrations of other molecules that it's affecting by binding to the receptor, say that it opens up a channel, affects something out?
Well, we're going to find that's certainly true. I'm not sure it's the most salient issue here, but although that does happen, yes. But,
It can, like, if you have too much of a hormone, like you get exposed too many times, you downregulate the number of receptors, and also like there can be like feedback mechanisms to make the receptor not do what it normally do.
all right. So William's talking about down regulation. We haven't talked about these ideas yet, but I do want you to sort of understand that as a human being, as a very complex physiological system, the longer the period of time that you are exposed to this hormone, the greater the concentration, your body eventually adapts. I wouldn't use so much refractory period as maybe what we were talking about earlier as a sensitization or habituation. The body simply adapts. And we talked about some types of receptors, sensory receptors adapting. We talked about them being fast or slow. Well, in this context, if there's no let up, if that hormone is at high concentration and remains at high concentration, as William suggests, the body is going to respond to maintain what? What's the whole objective of your
Homeostasis.
homeostasis? Excuse me. And so sometimes the body's successful, sometimes it's not. There's this issue of availability of needed cofactors or substrates. Yeah, sometimes we say that hormones work in concert with each other. Sometimes hormones and receptors require other molecules. For example, steroid receptors often require other hydrophobic compounds to bind to the DNA before they can do whatever it is they'll do. We'll talk about that later. Yeah, but this too is a determining factor. And then I guess what I was suggesting here with interaction with other hormones, we have some terms to describe this, and then you need to be aware that drugs can affect the activity of hormones as well. So again, I'll say this, but I can't tell you how many times folks forget this on a test. The same hormone can have different effects on different tissues. Why is that? Because the receptors can differ in those cells and tissues. So these are the terms that in this slide that we sometimes use to describe the relationship between various hormones. And at the very least, be familiar with these general definitions. Antagonistic, synergistic, and permissive. I think you can probably anticipate what we mean by antagonistic hormones. They simply have opposing physiological effects. For example, one hormone may increase the concentration of calcium in the blood. Another hormone may lower the concentration of calcium. One hormone may promote growth and development, cellular differentiation, and so on. Another hormone may act to inhibit that. So antagonistic hormones simply have more or less opposite or opposing effects. So they're not always complementary in that regard. Synergistic effects are a little bit more difficult to describe. If you look at the graph over here, the figure over here on the right, we have blood glucose concentration in milligrams per deciliter over time. All right, so what you'll notice in this figure is that cortisol, you just introduce cortisol into the patient, their blood glucose level increases a little bit, right, over time. Same thing with epinephrine, except it's even a more extensive response. But notice what happens when you combine glucagon, epinephrine, and cortisol. How would you describe the effect of all three relative to these? It's not simply additive, it exceeds the sum of the individual hormones. We call that synergy. So a synergistic response is sort of an amplification of the individual biological effects of those hormones. Two, three, or more hormones simply have a biological response that is either greater in magnitude or very different in quality than the individual hormones. A yet more subtle term to describe the relationship between hormones is the term permissive. And the idea behind the use of the term permissive is that a hormone only does its job well when it's in the presence of a second hormone. All right, so another hormone facilitates or optimizes the biological effects of a hormone. It's not that it's synergistic, it's just that in order to perform its biological function, that second hormone has to be present. As a classic example is vitamin D and parathyroid hormone. We've indicated that hormones typically circulate in the blood rather briefly. They have relatively short half-lives. We talked about the definition of half-life. I'll give you a table in just a moment that will summarize this. Do you have a question?
So is the permissive hormone the one that's in the presence of the other hormone, or is it the one that's being enhanced?
So you're asking, you're putting a teleological twist on this. You're saying that by that definition, one must have a purpose and the other, or one is more important, or?
No, you said in permissive, basically one hormone only does its job while in the presence of the
Correct.
other hormone. So which one's the permissive hormone? The one that
Oh, I see what you're saying. I don't know that that's always known. In other words, in many cases, let's say in the case of blood calcium regulation, PDH is the predominant hormone. So we say that vitamin D is permissive on PDH. But if you, in a rebellious mood, wanted to argue it the other way, I'd be perfectly open to that. The idea is that again, if the person sitting beside you is not in class when you come into class one day and you just feel very upset, you don't want to study, you want to pay attention, maybe she or he feels the same way about you. Right? Their study or their facility in the class may be dependent upon you. In some cases, it's not always clear. In the case of PDH and vitamin D, we would say that vitamin D is the permissive hormone because it potentiates the activity of PDH. And so, in many cases, I'm not sure that's always clear or even all that important. So as I say, I'll give you a table in just a moment that sort of summarizes the relative half-lives. But typically, most hormones have a half-life on the order of minutes to hours. The exceptions are thyroid hormone and the steroids. They tend to have somewhat longer half-lives. Now, we're going to talk a little bit about what happens when hormone levels are artificially increased for whatever reason, and that's part of your job, right? Part of your job is to artificially increase hormone levels in your patients. I've just found that to be a rule. That seems to be an all-purpose therapy these days. But there can be consequences to that. First of all, I want you to be familiar with the concept of cross-reactivity, because these hormones in some cases are peptides, or at least they bind to receptors. In many cases, they have very similar shapes. For example, insulin-like growth factor is similar to insulin, and thus they bind in some cases to the same receptor. So that's what we mean by cross-reactivity. If I artificially increase above normal levels the concentration of the hormone, it may start binding to receptors and have consequent physiological effects that under normal circumstances would not occur. And that can be unfortunate because normal metabolic pathways may be deranged as a consequence. They may go off track. And then if you look at this, I think this is what William was talking about before. This will be a recurring theme here. see this. We have one of the one of our objectives shortly will be to describe the majesty and joy of the hypothalamic pituitary axis, sometimes called hypothalamic pituitary target tissue axis. So you can see at the very top of our endocrine system here, we have the hypothalamus. The hypothalamus is going to release hormones that in turn cause the anterior pituitary to release hormones. Those anterior pituitary hormones then travel throughout the systemic circulation, for example, to bind to receptors in the thyroid gland. And then the thyroid hormone is released. All right. But as you look at this, and you can change whatever words you want here to make this clearer. But my question for you is, what are these red dashed lines in this illustration? Feedback, right. And we're going to be talking about the general nature of feedback. But what I want you to understand is that as thyroid hormone levels increase, it is important for you to realize that there are thyroid hormone receptors in your brain, in the pituitary gland and in the hypothalamus, that will detect the elevated concentration of thyroid hormone. Remember, what prompted the release of thyroid hormone to begin with? A hypothalamic hormone. So the idea here is that as thyroid hormones increase, that's going to trigger a reduction in the release of both the hypothalamic hormone and the pituitary hormone that were directly responsible for their release. And that's a type of feedback. And so what you get with too much circulating hormone is this inhibition feedback system, which in some cases can have adverse consequences. We'll talk about that a little later too. Remember we said that the duration of the hormone receptor interaction is very critical. Eventually, this whole system may just break down because of the very high concentration of the hormone in the system. Something some of you are also talking about upregulation and downregulation. This is what we're suggesting in the slide here. So one way that the body can maintain homeostasis in response to elevated hormones is by decreasing the biological response through a reduction in the number of receptors. So as the hormone level goes up, in order to maintain a consistent physiological response, the density or number of receptors may decrease. That's called receptor downregulation, and that's a very common mechanism by which tissues maintain homeostasis. However, notice the following exception. Initially, if the hormone level goes up, instead of causing a downregulation, there may be an initial upregulation. There may be an initial increase in the number of receptors. That's sometimes called priming or endocrine potentiation. And that will depend upon a variety of factors. It's generally transitory. And shortly thereafter, classic example is with insulin and regulation of glucose. There's rapid downregulation. All right, so this too is an important concept here. So if we go back to this illustration, I think I don't have a better one there. So what we're looking at again is the hypothalamic pituitary target tissue axis, sometimes called the HPA or the hypothalamic pituitary adrenal axis, or whatever the target tissue is. All right. The hypothalamus releases hormones that initiate this cascade of responses, but it doesn't release. Hypothalamic hormones are not released in a perfectly consistent fashion. Rather, for the remainder of our discussion of the endocrine system, I will be doing this. Because I want you to understand that the release of hypothalamic hormones is pulsatile. And when I say it's pulsatile, I mean in general in about a 90-minute cycle. So it goes up and then it goes down. It goes up and then it goes down. What do as you look at this illustration, as you look at this graphic, what do the hypothalamic hormones do? What is their effect? What is their purpose? to regulate the release of pituitary hormone. So what would you expect the pituitary hormone? How would you expect the levels of pituitary hormones to change over time? Yeah, it's also, under most circumstances, it's a little more complicated, but it also is going to be pulsatile after a little bit of a time lag. So be familiar with this pulsatility. And it even extends in many cases all the way down to the tertiary hormone, down to the what we're going to call the primary hormones. I should use the word tertiary in this context, because it's a different meaning. What are called the primary hormones. Primary hormones are the hormones that are released by the endocrine tissues of the body, not the brain. So, for example, the adrenal gland would be the products of the adrenal gland would be primary. The thyroid would be primary. The gonads would be primary. All right. So what we're saying is that the release of hormone from the primary tissues in many cases can also be pulsatile. So my question for you is why? Why would what's the advantage of this? What would be the purpose of pulsatile release based upon what we've talked about thus far? And I'll give you a clue. As a PA, you're going to be involved in administration of hormone replacement therapy. What does that mean? I'll give you a second. Look. What is hormone replacement therapy? How would you do it?
The body's not producing it anymore, so you're supplying it artificially.
All right, good, excellent response. So you would be supplying something that the body is not producing in normal amounts. Now, when you supply it, what is the method? I guess what I'm asking is, when you provide the replacement hormone to the patient, are you going to be providing it in this pulsatile fashion, or are you just going to give them a good shot and be done with it? Just put a patch on maybe there. But you would agree, whatever modality you as a PA use, it's not going to end up like this. And you know what? That has dramatic consequences. The fact that the mechanism that you use to provide the patient with the supplemental hormone is not pulsatile can dramatically change the physiology of that hormone. In some cases, we don't quite know why. In other cases, we just make up pretty much explanations on the basis of what we know about cell biology. And this will become clear when we talk about bone, for example. What is it? You might want to look up and see what one of the most effective therapies is for osteoporosis. It might be astounding. But in any case, that's my clue. So why would the hypothalamus release, I'm doing it already, why would the hypothalamus be releasing hormone in a pulsatile function? I think you're exactly right. Can you elaborate for the benefit of everyone what you mean by that?
If it's exposed to if it's realizing what the hormone's doing all the time, it won't have as great an effect. So if you were giving it artificially, the body will eventually not respond the way it would naturally.
Or if it does respond, the response may be momentary priming and then down regulation. So is it clear, Kelsey's correct, is it clear then that this vacillation, this oscillation, sort of keeps the receptors on their toes? Remember, what was one of the defining criteria of hormones in the blood? They are present in the blood in very minute concentrations, and it's that minute concentration that is varying. And these receptors that determine the biological response, are responding to very subtle changes in the concentration. But make sure you understand that if for any reason that hormone is artificially or perhaps even naturally elevated, the response, the physiological response, is going to be abnormal.
So we have to increase the dose over time then?
In some cases, but that's only going to work for a while.
Don't you normally check your blood levels? I mean, every time you do something that make sure that you're in the therapeutic threshold, and that's why you're monitoring constantly.
Well, I think that would be true regardless. That would not apply to just about any medication or any type of therapy. You have to make sure that what we're suggesting about the hormone is, and unless you can come up with the way, my guess is you will bring yourself great fame and fortune if you can come up with a way to release the hormone in a natural fashion as it is regulated from the hypothalamus, because you would certainly preclude a number of issues that you're going to deal with. This applies to reproductive hormones. It applies to, again, calcium regulation hormones. It applies to growth hormone. All of these things, when you provide them to the patients, you provide them at least in terms of their temporal, at least in terms of the way they're released into the body, in a very unnatural way.
But if we know the intervals for that, we can maybe mimic them, no?
I'm sorry?
If we know the intervals of that pulsatile secretion, maybe we can try to mimic it.
How? I agree with you, but
Well, if we know the intervals.
okay, well, let's say it's a 90-minute rhythm. Let's just say that it's a little, it actually is a little more complicated. Let's just say it is a 90-minute rhythm. What mechanism can you think of that would do, that, again, very, very tiny concentrations. We're not talking
pump
about,
that pumps every 90 minutes.
yeah, you can do a little bit with insulin, but
Right.
insulin is one of these hormones that is less sensitive than some others. Yeah, you're exactly right. You could, but would you propose? Think about injury to the pituitary gland. I don't want to get ahead here too fast, but most of these hormones, what we're talking about, are regulated by the pituitary, a tiny little tissue that is releasing a number of hormones simultaneously, each one of them responding to separate rhythms from the hypothalamus. Patient with pituitary damage, how are you going to sort all that out? How are you going to regulate each of those in a natural fashion? My only point here, my guess is that perhaps in your lifetime this will indeed happen, but I just want you to understand that right now, the replacement therapies often entail many adverse consequences, at least with respect to the natural cyclicity, and this is the reason why, because of this receptor dependence and the physiological response to the elevated concentrations. Another key point here when we're talking about pulsatile secretion, you need to appreciate that that's not the only fundamental rhythm of hormone release. All right, so the hypothalamus is releasing these, what we're going to call releasing hormones and inhibiting hormones in a pulsatile fashion. And for the purposes of this course, since it's a brief summer course, let's understand pulsatile to mean a 90-minute cycle, all right? But in addition to that, your hormones are subject to circadian cycles. What's a circadian cycle? Yeah, a change in the course of the 24-hour sort of solar day. For example, growth hormone peaks during night while you're asleep. ACTH, cortisol, also peak during the morning, for example. So there is a circadian rhythmicity to the release of hormones that is superimposed upon the pulsatile release. What do you know happens to hormone levels as you get older? Yeah. So there is a developmental cyclicity to hormones. When you're young, those hormones are going through the roof. Trust me, I vaguely remember that. And then as you approach adulthood, some of them increase. Most of you are over the hill now. You're shot too, so you can commiserate. And they start declining. So again, there is in some cases monthly, some cases seasonal. You've heard of seasonal affective disorder. Some hormones, some of the hormones regulated by pineal activity, just in front of the brainstem there. Some of those hormones are regulated seasonally. Others are regulated, as I say, developmentally. So I just want you to be aware that there is a profound and very very complex sequence of rhythms that regulate release of virtually all the hormones that we will be talking about. And you can see some examples here. So pay attention. You can see body temperature changing throughout the day over here. But here's the example of cortisol, and you can see it peaking about 9 o'clock there typically. Yeah.
Real quick before you get on classification, for circadian rhythms, like if you don't sleep at night for whatever reason, does that mean your hormones might have to be turned on, or?
Big time.
Okay. And then the other thing is,
Not only that, you're going to suffer greater frequency of accidents. You're not going to live as long. You have greater risk of cancer. You have greater risk of heart disease. We can just go right down the line. Yeah, we talked about it, but you're right, I didn't describe the proper term to it. So, excellent question. So what we mean by disuse atrophy is that, again, as far as I can tell, the primary function of healthcare workers is to artificially elevate hormone levels in patients. I'm joking, of course. In particular, you're going to be giving every patient for any possible malady cortisol or an analog. All right, so you're going to pump up your patients with cortisol. I know I feel a lot better when I'm pumped up with cortisol. So let's say we do that. Let's say that I have very severe inflammation, and you're giving me cortisol or prednisone or some analog to deal with that. All right, so what we're going to find, and we'll come back to disuse atrophy and talk about it in more detail. What we're going to find is that as you increase the concentration of primary hormone in my blood, how's the hypothalamus going to respond to that?
Negative feedback?
Tell the anterior pituitary to stop sending out hormone making hormone. That's the way of putting it. It's gonna stop making hormone. You're exactly right. So the hypothalamus is going to begin shutting down. The pituitary is going to begin shutting down. Normally, where does your cortisol come from? Anybody know? Your adrenal cortex. It's going to shut down too. So when we talk about disuse atrophy, what we mean is that, again, in these patients, you can have a transplant patient. I knew a fellow one time. My experience isn't trivial to what you folks will be going through shortly. And I knew a fellow one time who was a multi-organ transplant recipient. And he took just massive boluses of massive amounts of cortisol every day as a suppressant for his immune system. But he was extremely edematous. He had a number of problems associated with that anti-rejection therapy. But in particular, he had to take that hormone exogenously introduced from outside because his entire endocrine system had shut down. Disuse atrophy means that if there's an artificial or therapeutic, the term we're going to learn later is iatrogenic. If there is an artificial increase in the primary hormone level, your natural regulatory endocrine system no longer functions properly. It atrophies because from disuse, it's no longer needed, so it shuts down. Yeah.
I don't know if this is a weird question, but how does that work with like birth control when you're like when a girl's constantly giving herself these hormones?
Well, they have a little trick there. Again, we'll talk about this more, but in the contraceptives, in the little packet of pills, do all those pills have a pharmacological effect?
No.
In other words, you have a month of pills, but all of those have the same effect. About a week of those do not. They're just sort of placebo. So in part, that's intended to prevent this. But you're right. In some cases, for a variety of reasons, hormones may be more or less, what's the word I'm looking for, constitutive, the patches and so on. You look at the billboard up there, I'm supposed to be on a testosterone patch now. Anybody over the age of 36, as far as I can tell, is supposed to be on statins and a testosterone patch. But yes, under those circumstances, that would be true. That you would anticipate those and that's all I'm suggesting here. We're not getting into a discussion of deep pathophys here, but I want you to anticipate, even as we begin our discussion, that when you start playing around with the natural rhythmic release of hormones, there are going to be consequences. Sometimes you can use those for the benefit. As I say, look into the therapies for osteoporosis.
I was gonna say, are there permanent changes if you're on hormone therapy and then maybe you choose to stop, like with older people?
It all depends on the time. When we talk about disuse atrophy, the implication there is that changes are irreversible. But it depends on the interval. A lot of times it can be reversed if it's, if the patient is not on the supplemental hormone for a very long period of time. All right, so let's, as I say, I think this is the most important thing you'll get from our discussion of the endocrine system. Understand how the chemical classification of the hormones determines their function, binding, transport, and so on. I just think that's fundamental. Now, in reality, we can classify hormones in a variety of ways, and sometimes we'll do this. We're going to primarily focus on the structure. Remember when we talked about neurotransmitters, we talked about catecholamines. What do catecholamines have in common? Yeah, the precursor is tyrosine. So we can talk about chemical precursors. Serotonin is tryptophan, for example. Um, Catecholamines are also hormones. They're what we call neurocrine agents. They function both as neurotransmitters and hormones. But anyway, in any case, we can classify hormones by precursor. We can classify them by function. For example, some are mitotic. Some are not. By mitotic, we mean that they promote growth, cell division, and that can be useful. We classify them by mechanism. This will be important. So for example, some act as transcription factors. Others act through G protein pathways we've already talked about to do diverse things. We've already talked about the importance of solubility, right? We've said that some hormones are water soluble and other hormones are hydrophobic, and that has enormous implications for how they're transported in the blood, where the receptors are, and what they do for a living. So let's look at these structures. There's some good news and bad news. At the bottom, we have what are called the sort of the immunological hormones. These go by a variety of names here. They're called eicosanoids. We're not going to get into these, so we're going to cross these off our list just because the complexity would exceed the time that we have available. So we're not going to refer to the eicosanoids. That leaves us three classes here that we need to worry about. The steroids, and all of these are called steroids because they're derived from cholesterol, steroid. And you can see that would include everything from cortisol. We just talked about cortisol, estrogens, the male hormone testosterone, the fluid-regulating hormone aldosterone, vitamin D. This is vitamin D here, calcitriol. So you should know the steroids thoroughly. I would encourage you to just go ahead and memorize those. You just need to know those. We'll talk about these in more detail. There's actually two classes of these, but we won't worry about that now. Then there are these. We've already talked about these in our discussion of the neurotransmitter. We talked about the catecholamines. What are the catecholamines? Correct. Exactly right. Dopamine and the two adrenergics. And then also, we include in the amino acids, although that's probably not a good idea, the thyroid hormones. These are made up of a couple of amino acids that are fused together. Unfortunately, when you fuse them together, their chemical property changes. So the deal with the thyroid hormones is they are structurally what we're going to call amino acids, but they act like steroids. So thyroid hormones are placed into this structural class, but they function like steroids. So these two are easy, right? We learn these. There's only a handful of these. So if it's not a steroid, and it's not what we're going to call an amine or amino acid, it must be what? What type of hormone must it be? A peptide. So you can memorize the classes of the peptides up there. We're going to be talking about a lot of these, but basically by elimination. If the hormone is not a steroid or amino acid, it's got to be a peptide of some sort of. And those are going to be water soluble, as we'll see. And now in our cryptic little representation of a cell here, here's a cell. The yellow is the cytoplasm, the purple is the cell membrane, the yellow represents a receptor. I want you to notice a couple of things in this diagram about hormone receptor binding. First of all, in general, it will be the case that all hydrophilic hormones must bind at the cell membrane. They do not have the ability to gain access to the interior of the cell, so they bind to membrane receptors. Hydrophilic hormones bind to membrane receptors. As a general simplification, if the hormone has the ability, if the hormone is hydrophobic, i.e. steroids and thyroid hormone, exception everywhere, it has the ability to pass through the membrane and bind intracellularly. All right, so hydrophobic hormones can bind to intracellular receptors. Now, what do you notice about the location of these intracellular receptors? All right, it can either be in the cytoplasm or where are we here? In the nucleus. So make sure you make a note of that. Hydrophobic hormones bind, or let's state this more precisely. Hydrophobic hormones can bind intracellularly. Hydrophilic hormones cannot.
So hydrophobic can also bind membrane receptors.
Yeah, we'll get to that. But I just want to make sure you understand that generally they bind intracellularly to either a receptor in the cytoplasm or the nucleus. And then as pointed out, sometimes, and this is true, we're really finding a whole family, especially for estrogen receptors and aldosterone receptors, they too can bind in some cases at the cell membrane. So in some cases, hydrophobic hormones bind membrane receptors. They can go either way. Hydrophilic hormones can only bind at the membrane. Any questions about that? Now there's a little note over here about being more rapid and slower. That has more to do with the mechanism, how they operate, than anything else. I don't know that that's terribly important in this context. If you already know the answer to this, if the hormone binds at the membrane, what will be its general mechanism of action? You've seen this already several times.
Beta protein, G protein.
Doesn't do that. It does the G protein. And so this will almost always entail a G protein cascade or some similar cascade. As I say, I'm going to give you an oversimplified table of mechanisms in just a moment. And here's the thing to remember about what they're calling lipophilic or hydrophobic hormones, is that unless they bind at the membrane, they act as transcription factors. And the transcription factor alters the gene expression of the cell. Transcription factor alters gene expression. We don't have time to go beyond that. But look, just as we leave on this point, then make sure you understand what we're saying. What is the fundamental modus operandi of a steroid hormone or a thyroid hormone? To change the way that proteins are produced in the cells of the body. And thyroid hormone, for example, binds to about every cell in your body. Has broad spectrum metabolic effects. All right, we'll stop there and then be back for anatomy and